Now that we've covered the basics of the adaptive immune response and the cells that are involved in that response, we can finally get to the actual process of responding to an antigen. We're going to start this process with a new type of cell that we haven't really named yet, even though I've mentioned some cells that can act in this capacity. These cells are called antigen-presenting cells, or APCs. APCs can be cells that are infected with a specific pathogen, or they could be cells that are specialized to detect pathogens. In either case, they're going to detect that a pathogen is present and then show what's called the antigen fragment, a little piece of that pathogen, and they're going to they have a specific protein, you don't need to know what it's called, but think about it like a flagpole. And they can take this antigen and put it up on the flagpole and be like, hey, look at this thing I found. Everyone should be aware that I found this antigen. So these are antigen-presenting cells. They are going to somehow detect the presence of a pathogen and then present the antigen to surrounding cells so that the B cells and T cells can be aware of the presence of a specific pathogen. So looking at the figure, we have a pathogen right here. It is being ingested by this cell. So we can call this, for example, a phagocytic cell. And that phagocytic cell, as it breaks down that pathogen, is going to display a little bit of like a cell surface protein from that bacterial cell. And that is the antigen that it is presenting. That antigen can then be detected by B cells and T cells. So here we have a T cell detecting that antigen fragment from the antigen-presenting cell. So this process has to start with an APC presenting an antigen fragment. Once APCs are actively presenting antigen fragments, helper T cells are the next type of cell in line. So these are T cells that have the Ag receptors that are going to be compatible with the fragment presented by the APC. So here we have an APC, right? This is our APC. It's presenting the little orange upside down house. That is our antigen fragment. And here we have a helper T cell that is detecting it. The helper T cell is going to be activated in this process via cytokines. And we've talked about cytokines already in the context of an inflammatory response. They are local regulators. It's autocrine and paracrine signaling, so it's local regulators that are being released by, for example, phagocytic cells, other white blood cells, when they detect a pathogen. And the point is to recruit and activate other immune system components. So here we have paracrine signaling between the APC and the helper T cell. So that's cytokines activating the helper T cell. And then that helper T cell is going to release its own cytokines, which it is going to detect. So that's autocrine signaling. This is going to be positive feedback. We have the detection of the APC. That's going to trigger the release of the cytokines. So that's going to activate this cell, which is going to trigger the release of additional cytokines, which will amplify its activity. So it's a positive feedback loop of activation for the helper T cells. Once the helper T cells are active, they're going to subsequently activate other components of the adaptive immune response. So they're going to activate B cells, which are going to be involved in the humoral response, and they're going to activate cytotoxic T cells, which are going to be involved in the cell-mediated response. So now we'll look at each of these individual responses. So you can think of this sequence so far as we've got a flow chart, right, where we've got our APC, we've got our helper T cell that's activated, and now it branches, and we could do two things. We could do B cells, and we could do cytotoxic T cells, and those are two separate sets of responses that are both promoted by the active helper T cells. We're going to start with the cytotoxic T cells, which are responsible for the cell-mediated response. Basically, the cytotoxic T cells are very similar to the natural killer cells we talked about last time. They're going to go around and detect infected cells before the pathogens infecting them are able to mature. So if you have a virus infecting a cell, it's going to make thousands more viruses. If you can kill that host cell before those viruses are ready, then you've stopped that particular line of infection. That's what the cytotoxic T cells are doing. Cytotoxic T cells require two signals to be activated. First is you need to receive a signal from a helper T cell. So you need to have cytokines from a helper T cell activating the cytotoxic T cells. The second thing cytotoxic T cells need is interactions with an APC. So a cytotoxic T cell is only going to be activated if it's simultaneously detecting an antigen fragment from an APC and also detecting cytokines from a helper T cell. So you need both of those things to activate your cytotoxic T cells to trigger the cell-mediated adaptive immune response. Once those cytotoxic T cells are active, they can go around and detect cells that are infected with a particular pathogen. So in this flowchart right here, we have an infected cell that is acting as an APC. So what this cell would need, the cytotoxic T cell would need to do its thing, is a helper T cell saying, hey, it's time for you to activate, get some cytokines going in there, plus detecting that antigen fragment. Once it detects that antigen fragment and has been activated by the cytotoxic T cell, it's going to release two different things. One is perforin, which is a protein that's going to form pores in the plasma membrane of the infected cell. You can see that right here. That's going to disrupt the ability of that cell to function because it destroys the polarity of that membrane. So it's going to, for example, get rid of the sodium and potassium gradients because it's just a big hole in the membrane that allows things to diffuse through. It also releases a different set of enzymes called granzymes, which then get into that infected cell and trigger apoptosis. So this cell is going to go through its kind of automated cell death process. The important thing here is twofold. One, we're going to kill this infected cell, hopefully before the viruses infecting it mature and are able to burst out and infect other cells. And two, we're going to do this in an orderly way through apoptosis rather than like lysis, because when a cell lyses, it releases things that could potentially damage the surrounding tissue. But if you go through apoptosis, this programmed cell death, this is a much more orderly process, so the cell actually dies without damaging the surrounding cells. So that's the activity of cytotoxic T cells. Once that infected cell dies, that T cell is going to move on and search for other APCs that are infected cells that it can do the same process to. The other side of this adaptive immune response pathway is the humoral response, and that's going to be mediated by B cells and also antibodies. Like cytotoxic T cells, B cells also require two separate signals in order to be activated. First, they need to actually detect the pathogen. So we can either have APCs or antigen fragments just floating around, or the whole pathogen itself need to be binding to those AG receptors. The second thing we need is that the B cell is acting as an APC that is going to associate with an activated helper T cell. When the helper T cell detects this APC right here, the B cell acting as an APC, it'll release cytokines, and that will activate the B cell. So again, two things we need here. We need the antigen itself, and we need activated helper T cells, and that's going to then activate the B cell via a cytokine signal from the helper T. Once that B cell is active, it's going to divide very rapidly and generate two sets of cells. First will be the memory B cells that we're going to keep in reserve just in case we ever see this pathogen again. And the second thing is the plasma cells. These are the cells that are going to make antibodies, the soluble form of those B cell antigen receptors. And the antibodies are going to be things that are directly responsible for the humoral response. Antibodies have three main jobs. We're going to talk about two of them right here, and then the third one on the next slide because it's a part of a broader system. The big thing with antibodies is that they do not kill the pathogen directly. They operate through two direct mechanisms, though, to make it easier to get rid of the pathogen. For viruses, antibodies work via neutralization. The idea here is that those antibodies are compatible with the surface proteins of the virus, whether it's the capsid proteins or the proteins that are sticking through the envelope. Whatever proteins are sticking out of the surface of the virus, those are the things that have to interact with the receptors on your cells to allow the virus to infect your cells. Well, the antibodies are going to bind to those receptors and basically block them so that they can't interact with the receptors on your cells. That prevents the viruses from infecting your cells and allows your immune system to do the mop-up work. In terms of bacterial pathogens, antibodies work via opsonization. Opsonization is just binding to the antigens on the surface of the bacterial cells, and in doing so, promoting phagocytosis. Basically, our phagocytic cells recognize the back end of those antibodies. When they see those, they just gobble up whatever it's attached to. So if you can coat your bacteria in antibodies, your phagocytic cells, like this cell called a macrophage, you don't need to know that term, but it's something you may have heard in the context of your immune system. Your macrophages go around, when they see something coated in antibodies, they just gobble that thing right up because they know that is a pathogen that has been identified as such by your adaptive immune system. So again, antibodies are not killing the pathogens directly. They're either going to inactivate the viruses via neutralization or promote phagocytosis of bacteria via opsonization. The third thing that antibodies are involved with is the complement system, which is something I mentioned last time and said we were going to come back to, because the complement system is a really interesting mix of components of the adaptive immune response and the innate immune response. The way the complement system works is that antibodies bind to the antigen receptor on foreign cells, and this leads to a whole series of steps. Basically, there are proteins circulating in your blood that are part of this complement system that are going to bind the antibody-antigen complex, or ABAG complex. So right here, we have our ABAG complex, and here are the proteins of the complement system. So they're going to recognize, aha, there's, and I'm personifying these proteins a bit, they're not actually thinking this, but they just bind to things. They have an affinity for this ABAG complex, so when it's present on the surface of cells, they say, aha, they bind to it. That triggers the formation, and this is one of my favorite names in biology, that triggers the formation of pores in the plasma membrane of that bacterial cell called the membrane attack complex. That's what it's called. It's the membrane attack complex. And that basically tells you what it does. It puts holes in the plasma membranes of these invading cells, which causes water and ions to rush into that cell, lysing that cell. So that's how the complement system is going to kill pathogens. This is the third thing that antibodies are involved in. It's not something they do directly, but they are kind of a middle signaling step in this complement system. Those complement proteins, those are considered part of the innate immune system because they're not detecting specific pathogens, they're just detecting the presence of this ABAG complex. And going back a little bit further, when we talked about the contents of blood plasma and we talked about proteins that circulate in your blood, one of the categories of those proteins was things involved in the immune response, and the complement proteins are what I was talking about back then, even though I didn't name them. These are some of those blood proteins that circulate in the plasma. So that's the complement system. Isn't that awesome? I love the complement system. This is so cool. We're going to finish this section with two figures that kind of summarize the activity of the adaptive immune response. This first one is figure 43.20. In this figure, we've got the humoral response on the left and the cell-mediated on the right. It starts with an APC detecting an antigen that activates our helper T cells. So we have our positive feedback loop of helper T activation. And then remember, this is a branching flowchart. So on one side, we've got the cell-mediated response, where those cytotoxic T cells are going to get activated and go after infected cells in your body. And on the other side, we've got the B cells. Those are going to generate plasma cells to make antibodies. And also, ultimately, we're going to generate memory cells so that we can respond more robustly later on. This is figure 43.16, and it is the exact same information, just shown in a different way. What we see here is an APC detecting a pathogen and activating a helper T cell. So here we have the activation of the helper T cell via those cytokine signals. And then here we have the activated helper T cells activating B cells and cytotoxic T cells. Same information we saw in the previous figure, just shown in a different way. So I don't really care which way you like of showing it, but you should just know that pathway, APC to helper T cell, and then splits to both B cells and cytotoxic T cells. Know that pathway. So that wraps up our big look at the adaptive immune response. We're going to finish today by looking at the greatest invention ever, vaccines, which only work because of this system.